I apologize, I forgot to record that first part, but hopefully if you're joining in on just the video, you can see what we're doing here. And I will have this, of course, scanned and uploaded into your lectures module. Now, here is the problem with this reaction that is so important. The problem with this reaction is that it is not spontaneous. It has a delta G that is 16.3 kilojoules per mole. In other words, yes, it is positive. It is non-spontaneous. But what the enzyme does is it takes this reaction and it couples it with this transaction here where ATP and ADP are converted between each other. And so what it does is it's called a reaction coupling. And I'm going to use some abbreviations here. Okay, so we have F6P plus PO4 3 minus, giving us F16DP. I'm abbreviating these things because it's just too long. With a delta G of 16.3 kilojoules per mole. And what the enzyme does is it couples it with the hydrolysis of ATP. So ATP plus water. As a matter of fact, this also produces water here. That's a secondary product. And this generates ADP plus PO4, in other words, phosphate. This process has a delta G of negative 30.5 kilojoules per mole. And so when the enzyme that carries out this process combines those two reactions, you can see the phosphates cancel out, the water cancels out. So you end up with a net reaction, which is the fructose 6-phosphate plus ATP generating fructose 1,6-diphosphate, probably should put a parenthesis here, plus ADP. The combined delta G of that process is now minus 14.2 kilojoules per mole. In other words, it is now spontaneous. Whereas the original process, remember, was non-spontaneous. And so this is a trick that biological systems do very frequently. When they are faced with a step or a whole process that is thermodynamically non-spontaneous, the way they drive it through is by coupling that reaction with another reaction that is very spontaneous. And so the net combination of two generates a process that is spontaneous in the coupled reaction. The other thing is a side note. The enzymes that do these kinds of processes have an extra feature. And it is that they exhibit something that's called allosteric control. In other words, second site control. These enzymes function not so much like an on and off switch, but like a dimmer switch. And so many times, for example, once the product starts accumulating, it kind of retro feeds the enzyme and tells it, hey, slow down, we've got enough stuff made already. And so it controls the enzyme in much the same way through this feedback mechanism, in much the same way that a dimmer switch can control, let's say, the amount of light that comes from a lamp. So that is an interesting observation here. So just take it with you. All right. All right. Here is another interesting thing that I like also. Again, like I said, I am kind of like playing with my my tastes here, which is biochemistry. And let me show you this thing here. Let's see if we can get to see it. Okay. Oh, okay. Almost got it there. Yeah. All right. So enzymes, as well as many other components of cells, some of the components of membranes, some of the components of the structure that holds things together in the cell, are essentially proteins. And proteins consist of a chain of subunits called amino acids. Amino acids are substances that contain both an acid and an amine or basic group, as well as a distinguishing side chain that is what differentiates between one amino acid and another. Of course, you remember from your biology class that the genetic code in the DNA of the organism carries the information for the cell to make these proteins and basically put the amino acids in the proper sequence. Once the protein is released from the synthesis machinery, it begins to fold because some of these amino acids are able to interact with other amino acids in the chain. For example, negative with positively charged amino acids, or hydrogen bonding interactions, or nonpolar with nonpolar type of London dispersion, or also called hydrophobic interactions. And because these brings together amino acids that might be apart from each other on the primary sequence of the chain, it causes the protein chain to fold into what is called the native structure. So many of these enzymes that I was talking about are globular. In other words, they have like a more like a spherical shape. And this is what is called the native structure. And the native structure of a protein is typically the form that is active biologically. Notice that in order for the protein to exert its function, it has to be folded properly. Now, there are several influences, several stresses that can cause a protein to lose that active or native form, and we end up unfolding the protein and causing what is called a denatured form. So in its denatured form, let me zoom out a little bit here. Oops, not so much, no tanto. There you go. In its denatured form, of course, the protein is no longer going to be active. So one of the things that's interesting to study is what would be the effect of temperature on this process. So I looked up some information, and the reason, I'll be honest with you, the reason why I thought about this, because Tuesday I was talking to one of you who had their doctor's appointment and was sent home because they found the temperature was like, instead of 98.6, it was like 91 or something like that, which I thought was ridiculous and exaggerated. But it occurred to me to think, how high does the temperature have to be before proteins, let's say in an organism, start, kind of like breaking down, not breaking down in terms of chemically, but basically getting denatured. And you can imagine that if you increase the temperature, you are causing some of these molecules to vibrate, to roll around, to kind of like basically break up these intermolecular forces that are holding them together, and the excessive kinetic energy would cause it to denature or to unfold. So let's consider this as an equilibrium between the native and the denatured state. Looking up at some information, let's say that the overall heat involved in this process is 512 kilojoules per mole. Remember, you're breaking up intermolecular attraction forces. So we're not talking about breaking chemical bonds here. In other words, you're not talking in the thousands of kilojoules per mole, but it is fairly significant. And you can imagine why it would be positive, right? It would be positive because, right, you're breaking up interactions that were stable, so you had to pump energy in. Now, let's say that the entropy change for this process, I looked these numbers up. I forget where they came from, and I even forgot what protein specifically was. It was a small protein that had none of what are called secondary or post-translational modifications, where sometimes cells attach other non-amino acid things to the protein chain just to either make it more active or less active or whatever. So this is 1.60 kilojoules per mole. Does it make sense that the entropy change is positive? Yeah, right, because you are going from a highly ordered to a disordered kind of a structure, so it makes sense that it'll be positive, right? Okay, now, what would be the free energy change for this process? And let's just consider it in the standard state, okay? Let's not bother with other things. Well, remember the equation, right? Delta H minus T delta S. So that would be 512 kilojoules per mole minus the temperature. Let's say we're going to do this at body temperature, 37 degrees. How about that? Let's say at 37 degrees Celsius, because that's the average or supposedly the standard temperature of the body. Even though I was sharing this on Tuesday night with who I was talking to, that I think I remember reading last year, there was an article that came up where this concept of the, normal body temperature was totally, questioned, and saying, we should go back and look at that data and make sure we have it right. So it may be that we've had it wrong for all these years anyway, but it's okay. If it's off, it's not going to be off by that much. Okay, so 37 degrees Celsius is 310 Kelvin. And actually, remember that the entropy does have a per Kelvin unit in there, I forgot. So this would be times 1.60 kilojoules per mole per Kelvin. So that means that the Kelvins are going to cancel out. And that comes up to be, I'm going to say 16 kilojoules per mole. So what does that say about it? Right. In other words, the denaturation, the unfolding of this is non-spontaneous. And to that, I would put out a collective major sigh, like, woof, menos mal, like, thank goodness. Because can you imagine if that process were spontaneous? It would mean that our bodies would have to invest energy in keeping all of these enzymes and proteins in their native active state. No, it is more spontaneous for them. Once these proteins are formed, their tendency is to go towards the native state. They want to stay folded and active. So that's good for us. But remember, this is at body temperature, right? Now think about it. Let's think about this for a moment. We're saying that thermodynamics favors a state that is more organized. In other words, a state that has less entropy than the denatured state, right? In other words, the equilibrium favors the folded native state, which has, less entropy. Right? Why would that be? Well, right. It comes back to understanding what the second law says. Second law says it's about the entropy change of the universe. So we had to consider, yes, being in the native state has less entropy. It's more organized, it's more ordered. There's less microstates, right? But remember, we had to consider, well, what's happening to the surroundings, right? What happens to the entropy of the surroundings when I fold that protein around? I mean, what happens to the water molecules around it? Also, in the direction we're looking at, delta H is 512 kilojoules per mole, but delta H of the reverse process would be negative 512 kilojoules per mole. You are making so many of these intermolecular connections that the energy released might be enough to simply offset the lesser entropy of the native state. Right? Good. See, I knew you're thinking my way. Now I'm going to ask another question. This is where it gets interesting here. Okay, let me pull this guy out of here for a moment. And let's ask the question. Oops, where am I? Oh, there we go. At what temperature would denaturation, in other words, the unfolding of that protein, become spontaneous? Because remember, we just decided that at 37 degrees, no, the protein prefers to stay folded in its native active state, and that's good for us. Well, remember, what we said is that the turning point is the point at which in this case, the standard free energy change switches from positive to negative, right? So it'll be at delta G equals 0. But remember, this is delta H minus T delta S. So if we solve for this temperature at this turning point or crossroads, that would be delta H over delta S, right? It's just math. I'm just rearranging that expression there. And that would be, we said delta H was 512 kilojoules per mole. And delta S was 1.60 kilojoules per mole Kelvin, which means the kilojoules are going to cancel out. So the moles, Kelvins will, go up. And I get a value here of about 320. I'm going to round it up, Kelvin. Dr. D. Kelvin doesn't say much to us. My thermometer doesn't read that. Oh, of course. If you go to the doctor and they stick a thermometer in you, it's actually going to read something like 47 degrees Celsius. Normally, and until I heard this week, but normally my understanding has been that the fever threshold, how do you spell this? Threshold, sorry, is about 42 degrees Celsius. Now, a temperature of 42 Celsius is concerning. It's high. It's, hey, we got to take care of this right now, right? That's what a doctor will say. Now, look at that. 42 degrees is only 5 degrees Celsius from the temperature at which at least that protein that I'm talking about starts unfolding. Imagine if this is common for many proteins in the organism, that at these, in these ranges, in the, mid to low 40s, right? I'm sorry, mid to high 40s, you start seeing some of this denaturation of proteins. And here's the thing. We did an example of just a complete denaturation, right? We're just looking at a glob of, we looked at a glob of protein essentially becoming a free-ranging, random chain. But it doesn't take a lot of denaturation for an enzyme to start losing its activity. So even if these proteins don't fall apart, don't kind of like almost like melt away, they will definitely drop down in their level of activity. And now many of your functions in the organism can start getting compromised. All right. So I thought I'd give you that as an example. Like I said, I got these numbers for, I forget which protein specifically, and who knows that they're all the same. But you get the drift, right? You get the drift that this is a process that, number one, at the temperature that is considered normal for the human body, most enzymes, most proteins will be fine. They will stay on full. The temperature is not going to compromise their integrity. However, at a fairly high temperature, and we're not talking super high, we're just talking in the 40 degrees Celsius range, you start losing the activity and perhaps even the structural integrity of several proteins in the body. Anyway, I thought you'd enjoy that. I don't know. Like I said, not even sure where I got this information from in the end, but I hope you enjoyed that discussion. All right.